Welcome everyone, I'm Susie Valdez and I will be your moderator for today's event. Thank you for joining us for this presentation titled Adding Value to Food and Agriculture Waste, Immediate and Longer Term Applications. It's my pleasure to introduce our speaker today, Dr. Vincent Bulone, Director of the Research Center at the University of Adelaide, Adelaide Glycomics Research Consortium for Agriculture Project Development and Fight Food Waste Cooperative Research Center. For a complete biography on our speaker, please visit the tab at the top of your screen. If you have any questions that arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will address your questions following his presentation. Please join me now in welcoming Dr. Vincent Bulone. I will now turn the presentation over to him. Welcome, sir.
So welcome to my presentation. First, I would like to thank Dr. David Bradley and the Agilent team for inviting me to present some of our activities here in Adelaide, but also in my other lab in Sweden at the Royal Institute of Technology. So my presentation will focus on some key examples where we are using biomolecules for specific applications and the research that leads to these applications. But before the scientific details, I thought I should introduce myself briefly. I'm a biochemist by training, and I have been running research programs on carbohydrates from plants, algae, oomycetes, and fungi for about 30 years now. We are very strong in enzymology, carbohydrate enzymology, and applications derived from carbohydrates, but also solving the structure and the properties of complex polysaccharides. And the applications are in a whole range of sectors, biomaterials. I will illustrate some of this in my presentation, but also bioenergy. The biomass can be fermented, in particular the carbohydrates, into ethanol. Food sustainability, I will not discuss this in my presentation, but we are targeting cell wall biosynthesis in pathogenic microorganisms for disease control. Biorefinery is the exploitation of biomolecules, again from the biomass, through sequential extractions of compounds and their utilization for a number of applications. And a lot of what we do, especially in my other lab in Sweden, is derived from forest products, in particular cellulose. The research centers I'm leading at the moment are the Agricultural Product Development Research Consortium. This is a grant funded by the South Australian state government, focusing on converting agricultural waste into products with a higher value. I'm also the research director of the Fight Food Waste CRC, Cooperative Research Center, dealing also with food waste, but across the entire value chain, not only around material that doesn't leave the farm, which is the focus of the Agricultural Product Development Research Consortium. And for both these initiatives, Adelaide Glycomics is our partner laboratory with Agilent Technologies, plays a key role in analytical work for the different biomolecules we're analyzing. Look, this is the situation in Australia about the cost of agricultural waste. In Australia, it is estimated to represent around 20 billion Australian dollar per annum across the entire value chain, and 20% of this of the primary horticultural production is lost pre-farm gate, which represents close to 1.7, 1.8 billion loss per annum. But what is probably most worrying is the curves that we see on this slide. Since the mid-1990s, the population in Australia has increased by around 30%. And you can see what happens with the amount of waste we generate. It follows a much faster rate, and this is obviously unsustainable. So in our research programs, we are trying to address these issues by converting the waste into products with a higher value, making something useful. Food waste is a big problem, which is not specific to the Western countries, and the so-called developing countries also suffer from these issues. And everywhere in the world, basically, there is always a fraction of the population that suffers from malnutrition. And this again applies also in our so-called modern societies. If you take cauliflower, in this example, about only 15% of the cauliflower is actually eaten, and you can see that most of the rest is either left in the field, or it's non-edible, or it's simply wasted by the consumer. So we are trying to do something useful with the basically 85% that is not used, that is not eaten. At the moment, waste is used for three main applications. Either it is burnt to produce energy, and even the most efficient systems produce energy, but this is still a low value product, I would say. The biomass can also be composted or used for feed for animals. These different streams generate low value, $0 to $10 a ton if we're talking about feed for animals, for instance, and we think we can do much better than this. Is one of the activities of the CRC, which is about exploiting the waste in many different areas, but also trying to reduce the waste and change consumer behavior as well. So we have three programs in the CRC. The Reduce program, which is focusing, as the name infers, on reducing food waste. And this can be at different levels. For example, just simply optimizing packaging or ways to preserve food during packaging and transport is a way to reduce the food waste. The second program in which my activities are more specifically involved is the Transform program, where we are converting the biomass into a whole range of products. So this involves biorefinery, bioconversion, and then product development as well. And the third program, called Engage, is more about educating the future industry leaders, R&D leaders. So we have a program specifically dedicated to the training of PhD students in the food waste area in the broad context, but also trying to educate the end user, the consumer, to avoid and manage their food waste. And as you can see on the top of the slide, there are estimations on where the food is wasted across the entire value chain, and we are trying to act at every one of these different points represented at the top here to reduce at least by 50% the food waste in the next 10 years. So it's an ambitious program, but we are hoping to achieve at least some significant impact for society in that space. That I mentioned earlier at the beginning of the presentation is the state government grant called Research Consortium Program for Agricultural Product Development. So this is focusing on South Australian biomass, but we have multiple partners across the country, but also international partners as well. And without going into the details, the program one is about understanding the structure of complex carbohydrates and bioactives that we find in agricultural crops. Program 2 is about assessing their properties, either biological activity, and we're talking, for instance, about antifungal properties, anti-inflammatory properties, antioxidant properties, anti-cancer properties, but also the structural properties of carbohydrates like cellulose and starch and chitin from mushrooms. And program 3 is really about developing products from these different molecules from the waste and scaling up the production of some of the molecules and also exploiting them commercially eventually. What we are trying to do is to use as close to 100% of the waste as possible so that we do not generate waste from the waste, if I can say, by doing sequential extractions of the molecules and minimizing the residue that we will have at the end. There is no point targeting a molecule that is of super high value, if I can say, but that is present only at 1 to 2% in the biomass, because if we do that, we will still have 95-98% of waste. So we are trying to basically have an integrated sequential process that will extract a whole range of compounds sequentially and derive them into products with a higher value. And again, here, Adelaide Glycomics is playing a key role in providing analytical support through our partner laboratory with Agilent to these different. For the rest of my presentation, I decided to take some examples. I could go on for quite a while, but I picked a few examples to illustrate what we are doing more concretely. So the first polymer we're interested in is cellulose. Cellulose is present in every single plant at different levels, depending on the type of crop considered, but also the type of tissue. So it varies from about 20% to up to 80%, depending again on the type of tissues, and therefore it can be used as a valuable product from basically any type of waste, and this is what we are. First of all, we can prepare cellulose in different forms, as cellulose nanofibrils, abbreviated by CNF here, cellulose nanocrystals, abbreviated by CNC, and I will provide examples in a minute about how to use CNF and CNCs for applications. Another form of cellulose that does not come from plants is bacterial cellulose. It is, however, extremely expensive to produce at the moment, and it would be, even if it would become commercially viable, it would still be for a niche market because it's also difficult to use on a large scale. But the highest source of cellulose is, of course, wood. The problem with wood cellulose is that it's heavily lignified, which means that it needs to be processed, which is expensive and also generates a lot of solvent issues that need to be managed. But cellulose, as summarized on this slide, can be used for many different applications, and I'm going to give you a little bit more insight into that in the next slides. So if you would pull out from your shirt, your cotton shirt, a fiber, it would look like it is represented at the top of the slide. In other words, it's a highly aggregated network of cellulose microfibrils. And this network can be used, obviously, for textile and paper, but we can do more useful things if we dissociate this network. And there are two main ways to doing that. One is through mechanical shearing. The other one is by combining mechanical shearing with enzymatic hydrolysis. And when you do that, you basically dissociate the sub-elements of these macrofibrils into microfibrillated cellulose. And this microfibrillated cellulose can be visualized with different forms of microscopy, and it can be converted, for instance, in hydrogels that can be used for cosmetics applications, for instance, and other human applications. So it's quite a straightforward process. If you would make a nanopaper by simply drying the microfibrillated cellulose, you can derive the cellulose in a transparent film that is bendable and that can also be modified on the surface to make it, for instance, water repellent and design transparent self-cleaning surfaces. The reason why this material is transparent compared to normal paper is because it consists of microfibrillated cellulose. And as you can see on this slide, the pores of the nanopaper compared to a classical piece of paper are very small, which means that the light does not diffuse as much as in a normal paper, which creates opacity in the normal paper, whereas the nanopaper is instead transparent because it doesn't diffuse the light as much as this microfibrillated cellulose can also be freeze-dried to generate super tough foams. The material you see on this slide consists only of 30% solid material. The rest is air. and it's as light as polystyrene, so it can be used potentially for packaging as an insulating material because it's porous, but also as a carrier for pharmaceutical and biomedical applications, for instance. So it's super light and super tough, and it can be converted into a whole range of applications through a very simple process, again, dissociating the fibers and freeze drying them to generate. Another thing we can do with cellulose is to hydrolyze it. So this is how cellulose looks like if you extract it from a primary cell wall from a plant in a native form. But if you treat it with a strong acid, you will hydrolyze the least crystalline parts of the cellulose microfibrils and end up with what we call cellulose whiskers, because they are similar in their aspects as cat whiskers. This is where the name comes from. So these are basically the most crystalline parts of the cellulosic microfibrils, and they are quite homogeneous inside. They are in the range of 200 to 300 nanometers. So what can we do with these nanocrystals? Well, we can suspend them in a liquid, and depending on the concentration of the nanocrystals, we can potentially generate, or really generate, I should say, what is called a Chiral nematic phase, or if you like, it's a liquid crystalline phase. And this is what is represented at the bottom of the tube. If the sample is more diluted, as represented at the top of the tube, you end up with an isotropic suspension where the nanocrystals have a random orientation. But as you concentrate the suspension, again represented at the bottom of the tube, a liquid crystalline phase forms spontaneously. So if we look at what's happening in the bottom of the tube, from the bottom of the tube to the top, you can see that on each plane, the cellulose nanocrystals, which are represented by these small black lines, slightly change orientation by a small angle. And as you keep going up along the tube, the angle changes from one plane to another. And this is a spontaneous process. If you would dry this material, you end up with the films that are represented on the right-hand side that are iridescent. In other words, they interfere with the transport of the light through the material, or in other words, the color changes. The color of the material changes depending on the incidence of the light on the material. Inspired by nature, it's the same process as used by some insects. There are no pigments, in fact, involved here at all. It's just nanostructures. In case of the insects, it's nanostructures made of chitin that provide iridescence. So in this particular case, we have used cellulose, and if the nanocrystals are combined with salt, we can make colored films that again change color depending on the incidence of light. Examples of applications are in the paints, for instance. So you could have a paint for your car made of this material that changes color slightly depending on the incidence of the light from the sun on the paint of your car. And there are many other potential applications of this. In the other example, I said at the beginning we are quite strong in carbohydrate enzymology. I will show you a couple of examples of what we can do with enzymes that act on carbohydrate polymers. So here we have used an enzyme that acts on alginates. This is a carbohydrate that is found in seaweeds, and it consists of uronic acid, represented here in pink, and guluronic acid, represented in blue. The enzyme we have isolated acts faster on polymuronic acid stretches than it does on polyguluronic acid stretches. So if you incubate alginate from, let's say, brown seaweed, in the presence of this enzyme, it will release an unsaturated monosaccharide, and this unsaturated monosaccharide is spontaneously converted, it's a spontaneous reaction, into a molecule that we call DEH for deoxyerythroexoxylose uronic acid. And this molecule can be used as a precursor for bioplastics, but also potentially as a whitening agent for skin formulations, which has a lot of interest in some parts of the world. So expressed this enzyme and characterized it biochemically. We have shown that indeed it interacts more favorably with stretches of mannuronic acid than guluronic acids, and we have determined its pH, optimal temperature, and it's actually active on quite a large range of pH and temperature, which makes it very useful potentially for industrial. So when we use the enzyme on alginates, it degrades preferentially, again, the polymannuronic acid stretches to generate DEH, and this slide demonstrates that we are able to produce DEH in a rigorously pure form, and DEH is actually ethanol, which makes it very easy to purify. We have demonstrated that the process can be scaled up. This is what is represented on this slide. So when we add the enzyme, which is abbreviated here by TC-ALG1, to alginate in water, we can then simply incubate the enzyme with the alginate. It will release the DEH molecule, which is soluble in ethanol. So by simply adding 95% ethanol, we simply solubilize in one step the DEH molecule, and the rest remains insoluble, and the rest that is insoluble is the part of the alginate on which the enzyme has not acted. And this part of the alginate is rich in guluronic acid. Remember, the enzyme doesn't act very well on guluronic acid, and this allows us to solubilize the DEH in ethanol while retaining in an insoluble form a guluronic acid-rich alginate residue. The DEH can be used, as I said before, as a precursor for bioplastic, but also in skin whitening formulations, whereas guluronic acid as a polymer has been shown to have biological activity, antifungal activity, and it is used at the moment in clinical trials for the treatment of respiratory diseases against Aspergillus. So it has antifungal properties, but also other biological activities. It can be used for the prevention of the formation of biofilms, for instance. So a lot of potential applications. The system is fully scalable. We have produced here in a 50 milliliter reaction about 2 grams of DEH, and the rest is alginate. A third example is about, again, the use of another enzyme, which belongs to the lytic polysaccharide monooxygenase family. We have isolated this enzyme from a fungal pathogen of rice, Fusarium fujikuroi, and we have expressed this enzyme recombinantly and purified it to homogeneity. And this is what this slide illustrates here. We have first expressed it with a signal sequence peptide. That's the PLB sequence, which allows the enzyme to be secreted in the culture medium, and the signal peptide is removed by a peptidase from the bacterial strains in which we have expressed it. So once we have purified, we were able to use it to modify chitin, for instance. So what this enzyme does is that it breaks down the chitin chain by creating a carboxylic function at the reducing ends of the sugars that are modified, and it acts specifically on carbon 1 of the sugar residues. What the enzyme does, though, is that it does not completely degrade chitin, which is a big advantage here, because it can be used for surface modification only. In other words, we do not lose the crystalline part of the chitin. So we can keep chitin fibers intact while introducing carboxylic functions at the surface of the chitin, which can then be used to anchor a whole range of molecules to functionalize the surface of the chitin. In these examples, we have used you can see in the box at the bottom of the slide, fluorescein, FITC, fluorescein isothiocyanate as a modifying agent, and also a small peptide that consists of glycine, alanine, and leucine as an example. Of what can be done is represented here. So on the left-hand side, on panel A, you can see at the top the modification of chitin, which has become fluorescence with FITC, and that's panel A1. Panel A2 is the same picture of the commercial chitin, but in the bright field observation under the optical microscope. Panels 3 and 4 are just the control without the treatment with the enzyme. So you can see that the reaction is highly specific. On panel B, we have modified lobster shells directly with the chitin or fragment of a lobster shell. So panel 1 is the control, and 2 and 3 are the fluorescently modified lobster shells, which are rich in chitin. So the enzyme is working even on solid materials. So this is heterogeneous catalysis, if you like. It's a very efficient enzyme. On the right-hand side, the mass spectra reflect also the capacity to modify oligosaccharides with the same reaction and modify them with peptides. And this can be used to make bioactive oligosaccharides functionalized with peptides, for instance. So the reaction is very versatile on a whole range of molecules, and this is summarized here on this slide, where again, chitin is first functionalized with the enzyme, the LPMO, to introduce carboxylic functions at the surface of the material, followed then by the grafting of a whole range of compounds for functionalization. And you can see here a number of specific applications. In this presentation, which is about the generation of UV protective materials. What we have done here is to exploit small molecules called mycosporines and mycosporine-like amino acids. These molecules absorb UVA and UVB radiations, and they are normally produced by different organisms, algae in particular, but also some cyanobacteria. And the fish that thrive in tropical waters, like the Great Barrier Reef in Australia, for instance, acquire these mycosporines by eating algae, and it's a way for them to protect themselves against the negative impact of the UV lights. So what we have done in this was to exploit these mycosporines to generate UV protective materials. So there are two main types of mycosporines, which are derivatives of cycloheximine and cyclohexanone, and they're basically amino acid derivatives of these core structures. What is important on this slide is the values of the epsilon that you have at the bottom. These values reflect the molar absorption coefficient of these molecules, and it shows how powerful they are in absorbing ultraviolet radiations. These values are twice as high as what you need to claim that you have an efficient sunscreen product to be put on the market. So extremely high capacity to absorb UV radiation. So we have simply to make a conjugate with chitosan. So the molecules have carboxylic functions, and chitosan is a deacetylated form of chitin, which carries 3 amino groups. So by a simple amidation reaction, we can produce a stable conjugate, a covalent conjugate, that combines the properties of the chitosan with the UV absorbing properties of the mycosporines. And the conjugate can be turned into a very thin film of about 30 micrometer thick. The film is completely transparent, and it blocks completely UVA and UVB radiation. So unique properties here from this film. What is represented here is a modification of the chitosan by only 3% of mycosporines. If we increase the concentration to 10% of mycosporines, you can see that the UV radiations are completely blocked, whereas the transmittance in the visible light remains close to 100%. In other words, the films remain completely transparent while blocking completely UVA and UVB radiations. So here we have irradiated the films to an amount of UV radiations that is equivalent to the amount of sun you get over a period of nearly 10 years in Queensland and 15 years in Victoria and South Australia. And what this shows is that the film is stable under these high UV radiation conditions. In other words, it could be used as a coating material, for instance, for outdoor furniture, and still be stable after 10 years in Queensland and 15 years in South Australia and Victoria. So lots of potential. It's also thermoresistant, so if you cook it at close to 100 degrees for up to 12 hours, its absorption capacity does not change, and this applies to the different mycosporines we have tested. So the MGLI, the chinorine, and the P334 are all stable after conjugation to chitosan. The control, abbreviated here by CS, is the chitosan itself that has not been modified. So you can see that the absorption spectra of the three conjugates we have made with the three mycosporines are thermoresistant. It's biocompatible. Here we have grown skin cells on the films, and we have used a live dead assay to check the viability of the cells. So what happens here is that the cells turn red when they are dead, and in fact, when they are alive, they turn green. When the cells are alive, they are able to metabolize the fluorescent dye that we have used, which is normally red, and convert it into a green fluorescent dye. If the cells are dead, they are not able to metabolize the dye, which remains red, and therefore the cells that are dead are visible in red color. In other words, and in summary here, you can see that we have basically no cell death, which means that the cells can grow happily on this type of material, which shows their biocompatibility and direct potential application for in human tissues. So a lot of versatility. It shows that the cells stick very well to the film. On the left, it's the chitosan that has not been modified. On the right is the chitosan modified with the glycine mycosporine, and you can see that the cells spread very well on the material. So they are highly biocompatible, and they can be used for skincare products and protect the skin against the impact of UV radiation. So this has been now patented, and we are developing it as a commercial product, not only for skincare products, but also for coating materials, outdoor furniture, glasses, outdoor textiles, and many more materials. This is the end of my presentation. I hope you have enjoyed the types of applications we can generate. These are only a few examples. I could continue for quite a while, and I will be happy then to take questions. And here I'm also thanking, of course, the people that have contributed to the work, especially from my lab in Sweden. The names of the contributors are highlighted in green. And thank you for your attention, and I hope you enjoyed this presentation.
Thank you, Dr. Bulone, for that outstanding presentation. And we will now move to the live Q&A portion of the presentation. As a reminder, please submit your questions via the Q&A box. So we'll give them a couple seconds to get their questions in. Would you like to add any comments to our audience right now, Dr. Bulone, while we're waiting for some questions?
Yeah, a question that I'm very often asked, I don't know if this is going to come, is how far, we are from commercialization. It depends on the products, I would say. In most instances, we still need to upscale the production of some of these. So, for instance, for the UV protective materials, we are dependent on obtaining mycosporines on a larger scale. A way to do that is through metabolic engineering, so reconstructing the pathway, the biosynthetic pathways in organisms we can grow easily, like bacteria or yeast, to produce the molecules in large amounts. But basically, we have made formulations that work for skincare products, for instance. Other compounds that are dependent on the use of enzymes require, again, larger scale production of enzymes in fermenters, which can be done, of course. These enzymes are easy to express. We have demonstrated that. For the nanocellulose, it's already a commercial product, and it can be used in very many different applications. There's an annual growth rate, compound annual growth rate, predicted to be of around 30% for the next 5 to 10 years for nanocellulose. And most of it is done, it's already produced industrially, and most of it comes from wood. And it can go, I gave these examples here, but it can go also in composite materials to make reinforced materials for car panels, or even for aeroplane panels, making materials super light but strong at the same time. So some of these products could go on the market, in the near future. They're all at different stages, and the limitations are always, upscaling, and also, of course, looking at the market demand and the cost of production.
Dr. Bulone, I want to thank you for your presentation today and your important research. And I want to thank our audience members and remind them that there are still opportunities to ask questions that were submitted but not answered today by our speaker will be addressed via email. And this presentation will be available on demand viewing through September 2021. Please share it with your colleague who might have been interested in this topic. Thank you again for your participation, and I hope you will enjoy and join me for the next presenter, George Gonzalez. Thank you again, Dr. Bulone. And if you're not moving into the virtual studio, take care, be safe. Until next time, everyone, bye-bye.
Thank you, bye-bye.